So I want to begin my talk by showing you a recent picture of my group. So each student in this picture is working on a project related to functional DNA, functional RNA, and these projects differ in many details, but in some sense, all of them are trying to address this kind of fundamental question: what sort of functions can nucleic acids have? So the kind of old or traditional answer to this question was nucleic acids are good at storing and transferring genetic information, but that's more or less it. But we now appreciate that nucleic acids can do many other things, including form binding sites for ligands. These kind of molecules are called aptamers, and also to catalyze chemical reactions. So our group is interested in this more modern view of nucleic acids, and we're trying to learn more about the interesting and useful things that DNA and RNA can do. So we are particularly interested in a class of functional DNA molecules called deoxyribozymes. These are DNA molecules that catalyze reactions. They were discovered relatively recently in 1994. They're typically identified using a method called artificial evolution, directed evolution, in vitro selection, selects if you're working with aptamers. Deoxyribozymes can catalyze a wide range of reactions. And also, despite although they sometimes have a poor reputation as catalysts, there are some examples that can be remarkably efficient. So this DNA zyme shown on the left leaves RNA, and it does so at a diffusion controlled limit of the reaction of about 10 to the 9th per molar per minute. So what that means is every time this DNA zyme collides with its substrate, it cleaves it. So people sometimes call this sort of enzyme catalytically perfect. There are many examples of proteins that behave like this, but this shows that DNA can act as a catalytic perfect enzyme as well. So why are we interested? Well, DNA zymes offer a number of advantages relative to more traditional enzymes made of protein or even RNA. DNA can be chemically synthesized, and for the sizes we're working with, this is typically in a matter of hours. The cost of synthesis is quite low compared to both protein and RNA. DNA molecules are extremely stable, especially compared to RNA. Functional DNA molecules can typically be denatured and refolded many times without losing activity. This can be useful for some applications. And finally, DNA molecules can be improved and modified using the power of artificial evolution. So today I'm going to talk a bit about the efforts of our group to develop deoxyribozyme sensors. So when we think about this, we kind of break it down into two workflows. The first is making a deoxyribozyme that can generate some sort of signal, like light or fluorescence. And the second part of it is somehow conditioning this deoxyribozyme so it only makes a signal in the presence of the ligand of interest, or maybe in the absence of the ligand of interest. So today I'll talk about both of these branches of the workflow, and I'm going to start with making a signal. So making deoxyribozymes that generate some sort of useful signal. So one source of inspiration for these sorts of experiments came from chemiluminescent organisms like this firefly and protein enzymes that generate chemiluminescence like luciferase. Another source of inspiration came from fluorescent organisms like this beautiful jellyfish and intrinsically fluorescent proteins like GFP. So these kind of proteins are super cool. They're also very useful. And so these kind of considerations prompted us to ask, might it be possible to make DNA molecules that are generating these kind of signals? So the starting point was a series of interesting substrates, some of which are shown here. So in their phosphorylated forms, these molecules don't generate signals, but things change when the phosphate group is removed. This substrate here is called CP star, generates light when the phosphate is removed. This one down here is called 4-MEP, generates fluorescence when the phosphate is removed. Finally, this one here is called PNPP, and it generates a yellow product when the phosphate is removed. So our idea was to make a series of DNA enzymes that remove the phosphate groups from these substrates and therefore generate different kinds of signals. So the next step was to figure out a way to identify rare DNA molecules that catalyze these sorts of reactions. So to do this, we use the following artificial evolution protocol. You've heard a bit about this technique in the presentations today. So we started in this case with a large random sequence library containing about 10 to the 15 different sequences. So again, this is a gigantic number, a number we don't really encounter in ordinary life. This library was incubated with substrate, and the hope was that rare library members would be able to transfer the phosphate group from the substrate to their own 5' hydroxyl. And again, this is going to trigger production of a signal, which is ultimately what we want. The next question was to figure out how to fish out these phosphorylated molecules from the unreacted library. And we did this by ligation. And the important point here is the ligation reaction requires a 5' phosphate, so only the reactive library members get modified. We then purified these by pH because they're now longer than the unreacted library members. They're then cut from the gel, amplified by PCR. And finally, these DNA strands are separated to regenerate single-stranded material for the next round of selection. So my first graduate student, Katka Shvelova, managed to get this to work. She isolated a deoxyribozyme that generates a chemiluminescent product and renamed this supernova. Here is the workflow of the light-producing reaction. You can see it's quite straightforward. You simply mix supernova with substrate and put it into a plate reader. Here is a secondary structure model of supernova based on a method called comparative sequence analysis. You can see that this is quite small, 46 nucleotides, which means it's easy and cheap to make. Here is the time course of the light-producing reaction. So you can see that the signal is visible very quickly, and under optimal conditions, we can now achieve signal-to-noise ratios of more than 10,000. A second talented graduate student in the group named Martin Volek used a similar approach to generate a deoxyribozyme that produces a fluorescent product. Here is the time course of the fluorescent reaction. There is a secondary structure model, again, based on comparative sequence analysis. And Martin was also able to make a fluorescent sensor based on the deoxyribozyme that can detect ribonuclease activity. I'm not going to talk about this now, but I will later in the talk, so keep it in the back of your mind. Martin was also able to make a third deoxyribozyme that generates a yellow product that can be seen by eye. So you can see it in this photograph. The yellow product is only visible when both deoxyribozyme and substrate are present. So to summarize, we've made 3 different signaling deoxyribozymes. Each one has a distinct secondary structure, uses a distinct substrate, and generates a different sort of signal. So at this point, we're trying to improve these deoxyribozymes in various ways. I don't really have time to talk about these projects in detail, but I just want to mention one highlight. So this is the fastest deoxyribozyme that we've isolated so far. This was developed by Katka, who was a grad student and now is a postdoc in the group. So you can see that this reaction finishes fairly quickly. The K observed is about 1.4 per minute. One exciting thing is this was done under subsaturating conditions, and if we increase the substrate concentration, eventually the rate gets too fast to measure by manual pipetting. So for us, this was exciting. One other point I want to make. This wasn't so easy. So here is the result of Katka's variant of supernova after her first selection, after her second selection, and after selections 3 and 4. This was about 1 PhD. So this is, I would say, a long and winding road. Okay, so I've talked a bit about making signals using deoxyribozymes, and now I'm going to talk a bit about modifying these deoxyribozymes to act as sensors. So there are different sort of ways to think about this, and most of our approaches attempt to make sensors that don't require washes or purification. So one way you can think about this is to engineer a deoxyribozyme in such a way that it is inactive unless some input molecule is present, and this somehow activates the deoxyribozyme. So this can be done in principle in many different ways. We're exploring a number of approaches in the group. Today, I will tell you about just one of them. So the goal of this project was to make a fluorescent sensor that can be used to detect ribonuclease activity. And the kind of key point here is all of our deoxyribozymes react with their 5' hydroxyl group. So if we covalently modify this with an RNA linkage, we can turn the deoxyribozyme off. If we add this to a sample that contains ribonuclease, this RNA linkage should be cleaved, which will restore the 5' OH and then regenerate catalytic activity. So that's the very simple idea of this design. Here's an experiment showing that this works. So the fluorescence signal is quite low in the absence of ribonuclease, quite a bit higher in the presence of ribonuclease. And we were also able to show that if we add a mixture of ribonuclease and ribonuclease inhibitor, we don't get any sort of activity. So this provides additional evidence in support of our mechanism, but it also suggests that maybe we can use our sensor to identify novel ribonuclease inhibitors. So one other point I want to make is because most ribonucleases catalyze multiple turnover reactions, a single RNase should be able to cleave and activate many copies of the sensor and therefore amplify the signal. So here's how we envision this working. The ribonuclease cleaves the sensor to restore the 5' OH. This then reacts with 4-MUP to generate this fluorescent product. The ribonuclease then diffuses away, does it again and again and again. This graph at the bottom shows the detection limit of our sensor. So in this case, we were able to detect concentrations of RNase A as low as about 100 picomolar. And again, this is a homogeneous assay that doesn't require washes or purifications. So the next question was whether we could actually use the sensor to address some sort of real world application. So what we decided to do is try to identify small molecules that inhibit a ribonuclease from COVID called NSP15 in the high throughput screen. So this shows how the workflow works. We start with our sensor in the off conformation. So again, it's turned off by the single RNA linkage. We mix it with substrate and aliquot this into the wells of 96 well plates. So each well contains NSP15, which is the protein we want to inhibit, and also a small molecule from a small molecule library provided by my friend and collaborator, Václav Vaverka. So the idea is if the small molecule is an inhibitor of NSP15, as shown on the left, NSP15 will not be able to cleave and activate our sensor, and therefore no fluorescence is generated. On the other hand, if the compound is not an inhibitor, we expect NSP15 to be able to cleave our sensor and therefore generate a signal. And because we expected that most of these small molecules would not be inhibitors, we thought we would see a series of plates that look like this with many fluorescent wells, and we're looking for these rare cases in which fluorescence was somehow reduced or quenched. So here's a result of the high throughput screening. So on the x-axis, I'm showing each of the 1,000 compounds in this library. They're arbitrarily numbered. And on the y-axis, I'm showing the fluorescence in the corresponding well. So you can see that in most of these wells, there's a strong fluorescent signal. We also see a handful of wells in which fluorescence is reduced. And we were able to show using independent assays that these molecules are in fact inhibitors. Here's one example. So this was exciting for us because it showed that our sensors can actually be used for some sort of real world application. So we've made a few other sensors. I don't have time to go into them in detail. I will briefly mention a few of the highlights. This first one detects oligonucleotides with specific sequences, and we can program it to detect more or less any oligo. The second one detects DNase activity. The third one we've already heard about detects RNase activity. This final one here detects an interesting protein called ApoBac3A. This is a protein that edits DNA and has been linked to cancers. So we think this assay could be useful for high throughput screening. I also want to point out that the limit of detection of these assays is reasonable, from 100 nanomolar to 100 picomolar. And again, all of these are homogeneous assays that don't require washes or purification. So from our point of view, one appealing aspect of these kind of assays is the workflow is quite simple. You simply mix the deoxyribozyme with substrate and put it into a plate reader. Also, these reactions are quite cheap. So This graph shows the signal to noise ratio we can achieve using supernova at different reaction costs. And we modulated this by increasing the DNA concentration. So we can get signal to noise ratios of about 1,000 at a cost of less than 0.1 euro per reaction. And this is cheaper than comparable methods like molecular beacons, was shown in the graph. Okay, so to summarize, we made 3 different deoxyribozymes that generate different kinds of signals. We've also developed a series of homogeneous sensors using these deoxyribozymes. And finally, we've shown that these sensors can be used in high-throughput screening. So some of this work was published in the summer in the journal Nucleic Acids Research, and it was also featured on the cover. So if you're interested in learning more, I would encourage you to check out the back-to-back papers by Martin Bolek et al. Okay, so finally, I would like to thank my group for their dedication and hard work. I would like to thank collaborators, particularly Rob for his high-throughput screening, funding agencies, the organizers for giving me a chance to talk, and finally, all of you for your attendance and hopefully attention. Thank you.